The Cell Cycle by Gwace and Twissas. The first part of the cell cycle is mitosis, and there are 5 stages of it. The first one is interphase. The first part of interphase is the G1 phase, where the cell brings metabolic activities to get ready for the S phase. In the S phase, the synthesis phase, synthesis means to make, and here we are making a DNA copy. Lastly is the G2 phase. Here is where organelles and molecules needed for cell division are made, and this gets the whole cell ready for mitosis. Next is prophase. In the first part, sister chromatids are formed by the DNA being compressed and made visible. These chromosomes are staying together by centromeres. Then the nuclear membrane breaks down. Lastly, the centrioles then go to the counterpoles and spindles fibers are made. Metaphase begins when spindle fibers attach the centromeres of all the chromosomes to their poles. Then the chromosomes are set in a straight line across the center of the cell, also known as the equator. The centromere in the middle of the chromosome is broken. Then the chromatid pair separates and is now known as daughter chromosomes. Then the spindle fibers bring a single chromatid to its side of the pole by trimming and thickening. Next is telophase, and this is the last one. Chromosomes join at the end of the cell and start to loosen. Nuclear membrane begins to reform. And lastly, the spindle fibers break down. In cytokinesis, the cell separates completely, so there's a space in between the two. At first, there is a chromatid inside the nucleus, and then it converts to chromatin, and the cycle is done, and you have 2 cells.